Your body is reacting to subtle frequencies beyond your hearing. We usually think of biological signals as chemical or electrical pulses, but what if minute mechanical inputs change everything happening inside? Imagine a single cell like an incredibly fragile tuning fork. Even the softest ripple from outside can shift its resonance enough to alter how it produces energy. This is not just theoretical noise, it involves real physical interactions at the cellular level. We are now studying phononic exchange modulations, which is simply the name for those tiny vibrational inputs influencing intrinsic cellular resonance and mitochondrial function stability. It suggests that perfect physiological coherence might be found not through chemicals, but through precise rhythmic patterns we can feel. The real question is not whether tiny inputs affect one cell. It is how those vibrations coordinate the entire system. Think of your body as an orchestra, usually playing at millions of discordant tempos. We are seeing evidence that precise rhythmic input acts like a conductor's baton. It synchronizes disparate rhythms across neural networks and physiological functions. This process, called neural entrainment through oscillatory input synchronization, allows external pacing to influence autonomic balance without triggering any specific chemical signal inside the brain. It is turning chaotic internal noise into coordinated flow. The effect does not just stop at coordinated flow, it goes deeper, right down into the scaffolding itself. Imagine your body not as soft tissue, but as an incredibly complex suspension bridge reacting to wind stress. That is what we are looking at when vibrations influence the extracellular matrix. These subtle physical stresses activate mechanical signaling pathways deep within the cell structure, a process called mechanotransduction. It means external patterns are not just nudging electrical signals. They are physically shifting how internal chemistry operates. This changes cellular function via its structural integrity, which means something nobody expected for how we perceive pain or stress. We established that mechanical inputs are fundamentally altering cellular structure, which changes function itself. But what if this is not just about static structural damage or improvement? What if these ultra-subtle vibrations can actually tune fundamental biological oscillators in your body, your heart rate for instance, without any chemical messenger being involved? We are talking about influencing intrinsic cardiac coherence through pure mechanical resonance, like tuning a bell with an outside hum. It is the theoretical limit of how physical stress dictates biochemical flow inside the cell. Yet many critics argue these subtle field state changes are just statistical noise and lack objective biomarkers to prove it happened non-invasively, a point researcher, Dr. Anya Sharma, vehemently disputes. But if they can measure that shift, everything we thought about systemic function is wrong. We know physical inputs influence cellular mechanics and rhythms. The next frontier is not just proving this, it is mapping it. We need advanced models to predict how a specific vibration pattern might stabilize a particular cell state before we even intervene, like designing an ultra-precise biological tuning mechanism that is far more intricate than anything available now. Think of trying to tune a symphony not by adjusting one instrument at a time, but by changing the entire acoustic environment. That is what these resonant frequency interactions are asking us to model. The cellular structures receive this vibrational pattern, but whether it triggers true self-correction or merely temporary stress response is dependent on something else entirely, and that factor changes everything about how we approach human biology. The answer is more complex than just repair or stress. If these vibrations truly guide intrinsic biological coherence, if they allow your system to resonate toward maximum efficiency, then we are not studying a physical reaction at all. We are observing an optimization process in real time. Imagine trying to fine-tune a million interconnected microprocessors with only general feedback. The current scientific methods cannot capture that predictive relationship between subtle input and perfect internal stability. We need to build a dynamic map, a sophisticated model of how those resonant frequencies interface before any physical change occurs inside the cell. But what if that inherent ability to self-optimize is far more sensitive than we give it credit for? The verdict is this subtle influence is not just noise. It is a signal for self-correction. If your biology possesses that innate ability to optimize itself through resonance, then the focus shifts entirely from repairing damage to guiding perfection. Think of tuning an instrument not by forcing notes but by finding its natural harmonic center. That is the ultimate goal here. This research points toward designing feedback mechanisms that use precise rhythmic patterning to gently nudge your body back into deep, self-sustaining coherence. Start experimenting with highly specific environmental inputs. Look for patterns that feel restorative rather than stimulating. The full research archive is at jmthomasofficial.com. The core lesson here is predictive resonance, not just reaction. We are moving beyond asking what happens after a vibration hits the cell. We must learn how to shape the vibrational input perfectly so that coherence begins before physical change occurs. Think of it like an architect designing a perfect frequency for an instrument, not hitting random notes, but finding its absolute natural pitch beforehand. This is where next-generation biofeedback tools become crucial, allowing you to model those micro patterns in real time. You are not fixing damage. You are optimizing the system's inherent operating code through subtle rhythmic input patterning. The full research archive is at jmthomasofficial.com.